So there are many, many neurotransmitters that are being released in our brains, and there are many ways that we can sort of divide them up and sort of classify them. But one way to sort of think about these neurotransmitters are, in terms of when they were identified or discovered. So, some of the neurotransmitters that we have a lot of familiarity with, I mean, like people have actually heard of the names of these before, like dopamine or acetylcholine or, norepinephrine or serotonin. They actually were identified, by neuroscientists rather early, historically, in the field of academic neuroscience. So these are actually referred to as classical neurotransmitters. Classical just means that they were identified or discovered early. And they include, of course, the most classical neurotransmitter of all, which is acetylcholine, which was the Vega stuff, right, that Otto Lowy, the Austrian, scientist, explored and investigated when he was, to some extent demonstrating the chemical nature of neuronal transmission. He with that experiment with the frog hearts and the vagus nerve. Another set of classical neurotransmitters are what we call the monoamine neurotransmitters, and the monoamines have one monoamine group, on each of them. So there's a chemical, feature that sort of ties them together. That monoamine itself comes from the amino acids, that are the raw materials for their synthesis. So, there are two monoamine neurotransmitters, dopamine and norepinephrine, that are both manufactured from the same amino acid. And that's an amino acid called tyrosine, which has a single, one monoamine group, like all the monoamines. But it also has a very distinctive kind of ring structure. It's called a catechol ring. So dopamine and norepinephrine are monoamines. They're made from amino acids, and they have one amine group. But because they're made from the same amino acid, tyrosine, that has this distinctive catechol ring, they're also a subcategory of monoamines. They're called the catecholamines. So dopamine and norepinephrine, are classical neurotransmitters. They're monoamines, and we're going to see that they're also catecholamines. While serotonin is another monoamine neurotransmitter. It's made from an amino acid, but it's made from a different amino acid, not tyrosine, but an amino acid called tryptophan. And that has a different ring structure. It's got an indole ring. So serotonin is sometimes called an indolamine. And what's interesting about that is that, neurotransmitters, these chemicals, will interact with receptors, right? And, there's some similarities amongst the catecholamines. They're closer to each other in terms of structure, than the catecholamines, dopamine and norepinephrine are to, another monoamine like serotonin. So sometimes in terms of drug development, this is, for another class in psychopharmacology, but, that those structural similarities are important in terms of designing drugs that can act, at receptors maybe for more than one monoamine neurotransmitter. So classical neurotransmitters, acetylcholine, the monoamines, including the catecholamines, dopamine and norepinephrine, and, the indolamine, serotonin. And then there are a class of, classical neurotransmitters that are actually far more abundant than the monoamines. They carry out the bulk of fast excitatory and fast inhibitory neurotransmission in our brains. These are the amino acid neurotransmitters, and they're actually very close to, amino acids. They include glutamate, which is our primary excitatory neurotransmitter in the brain, and then GABA, which is our primary inhibitory neurotransmitter in the brain. And then we have a whole raft of, newly identified or more recently identified neurotransmitters, non-classical neurotransmitters, including peptide neurotransmitters. These are short, proteins, but typically in a neuron that's going to release a neuropeptide neurotransmitter, it'll express a gene that'll build a bigger protein, that'll translate into a bigger protein, but it'll also express genes for, enzymes that will come and then cleave or cut out, smaller, short protein segments that will actually be, the peptide neurotransmitters. And, there are, for example, a lot of opioid neurotransmitters are actually short peptides, short proteins. like endorphins or enkephalins, endogenous opioids, that you release, to act at receptors that can help to, relieve pain, for example. Then there were also lipid neurotransmitters. These are non-classical transmitters that are absolutely fascinating. There's one called anandamide, another called 2-AG. These are cannabinoid, endogenous cannabinoid neurotransmitters. These act at receptors where the phytocannabinoids, the plant-derived, chemicals in cannabis, actually act in your own brains. And they have a really interesting, kind of pharmacology. They're actually, we don't have time in this class. In the psychopharmacology class, we go into a lot more detail on the endocannabinoids and the phytocannabinoids, but they're actually, cleaved out of the membrane postsynaptically, typically, and then they work their way back, and act at receptors on the presynaptic neuron. So they're really unusual, these non-classical, neurotransmitters. One of the things about a lipid, right, as a neurotransmitter, is that you can't store it in a phospholipid bilayer bound vesicle because it will just leak out, right? So it's actually got some really interesting pharmacology. So anyway, classical neurotransmitters, those that were identified early, historically, and non-classical were identified more recently, and there's a lot of fascinating, complexity in these neurotransmitter systems at synapses.